Hi, good afternoon. Good afternoon. Welcome to seminar. We need to get started. Hey, it's great to see so many folks
I don't
here.
think you...
Yeah, right. Hey. Good afternoon. Thank you for coming. It's great to see a good turnout. For the third time this semester, we have went to great expense to bring you an internationally born speaker, right? So we're back to Australia. So this is Dr. Ronnie Baker. He's probably the newest faculty member at South, right, in marine science. So Ronnie did his undergraduate honors and PhD from James Cook University in Australia. Despite his funny accent, he was actually born in Hershey, Pennsylvania. But he lived with his Aussie family doing Aussie things in Australia until returning to the U.S. for the first time in 32 years. So I didn't write this, Ronnie did, and it's funny. But I promised him I'd work on the delivery to make it funny, and then if it bombed, he was going to tell you guys that I came up with this. So it's going well so far, so good job. All right. So anyway, after 32 years, he left Australia, came back to NOAA to work with Tom and Noah over at Galveston doing NOAA fisheries stuff. Did a lot of things. He met his wife, Jen, there, who some of you may have met. He has two kids, Finley and Julian. He actually did a postdoc at the Smithsonian following Jen there in Florida, where he postdoced with Candy Feller. And Candy will be here doing mangrove talks and stuff in April. So
April 12th.
April 12th. It'll be a good seminar. After the Smithsonian, he returned to Australia to be sure both of his children would be born there, so they would actually be Aussie raised and citizens. And then today he's going to talk about some of his work from Australia. So you guys help me welcome Ronnie.
Thank you. So today I'm going to talk to you about some of the work I was doing while I was back in Australia, but the work was actually in Papua New Guinea. So first I wanted just to start off by acknowledging the key players on the project I was working on. Jacob Wani and Marcus Sheaves were the project leaders. Marcus put in a lot of work over many years to get Jacob and the Federal Fisheries Agency there on board for this work. And then we had a really big team of people. These are just the researchers I worked with mostly. So our project was about sustainable management of a sport fishery in New Guinea for the benefit of local communities. There's some fishery species there that are fairly unique to the place. And the federal government realised that while remote area tourism sport fishing is not likely to be a major part of the national economy, it could be a very significant part of local communities where it occurs. And that through careful management and conservation of the resource and sustainable use, it could generate significant direct benefits for those communities, which in turn would promote conservation and stewardship, essentially using natural resources in a non-extractive manner, as opposed to the alternatives. And I like this diagram because not only does it sum up what we did, including biology and ecology, we had social scientists, economists, people involved in business, all working on the project, but it also shows for me the way that the sort of things I do, looking at juvenile habitat and such, can actually directly feed in and influence the lives of people. So where is New Guinea? I came from around here in Australia, Townsville on the east coast. It's actually part of the ocean right at the moment, but New Guinea is an island nation to the north. For people, history buffs, World War II interest, the capital, Port Moresby, is here. There was some fairly small scale but significant fighting along the Kokoda Track up in some pretty horrendous conditions. Some people may have heard of the Battle of Milne Bay, where forces came head to head, and the Battle of the Coral Sea occurred out here. In New Britain, you also have fairly significant events happening around Rabaul. So Rabaul was held by the Allies, captured by the Japanese, fairly significant base, and then was effectively isolated as the Allies swept back through the Pacific. What do we got? The work, our study site was on the island of New Britain up here. So New Guinea, Papua New Guinea is sort of the eastern end of the island of New Guinea. New Britain New Ireland, other islands. This is Bougainville and then the Solomons out here. And New Guinea, it's a really incredible place. There's something like 800 living languages. So a tiny fraction of the world's population, but a really large fraction of the world's languages. It's a really diverse place. And the majority of the population still live a relatively traditional lifestyle in village communities. They're not completely isolated by any means. In fact, they were even through World War II in some of the highland valleys. But the people rely very heavily on natural resources from their surrounding environment. In terms of industry, major economic development in the country, there's not many, and they tend to be this sort of thing. This is actually quite devastating. This, one of our study rivers, complete pristine forest, and they've just reached it, logging it, and although you can't see much of it here, there's large areas where there's just no riparian, just bulldoze right through. These logs will get loaded, they'll push through to the beach, bring barges in, load the logs, take them straight overseas. So local people get very little. A few will get employed during the logging enterprise. Some of them will die, get squashed by logs. But generally, the people will be left with a mess, and there'll be no lasting benefits. What typically follows these sort of logging enterprises is large-scale plantation agriculture. Some people have probably heard of palm oil. This is an oil palm plantation. It's about a middle-aged plantation. Compared to, say, equivalent stuff in Australia, sugarcane, I think this is actually nowhere near as destructive. And in New Guinea, it's not bulldozing orangutan habitat to grow the stuff, but on a large scale, it without any sort of management guidelines, it just dominates the entire seascape or the landscape. You can see here, no riparian left at all. Trees overhanging the beach, overhanging rivers, and you can see the sediment just gradually smothering these coastal coral reefs. But there are aspects of this style of agriculture, if it was managed properly, that actually lend itself very well to the way the society is structured in New Guinea. The other big money maker for the nation is mining. This is one of Australia's largest companies, going into the top of the highlands in New Guinea and open cut mine for gold. Now, if you're on the top of a really steep mountain range in a seismically active area that gets about 10 metres of rain, it's about 30-something feet of rain per year, you build a tailings dam, but what happens? It just goes poof. Then you start paying fines, official and unofficial, to the federal officials, and you just dump the tailings straight down the river. But this together with selling tuna fishery rights are the big sort of money spinners for the nation. So in these communities, though, remote communities where the sport fishing operations, there's not many, there's very few in fact, but this is an alternate, and the government looked at this as a possible alternate way that remote communities can get some economic development, help get their kids to school without relying on these large-scale extractive industries. So this is at the village we worked in. This is the entire school came over one day to come and have a look at what we were doing, look down the microscope and learn a bit more. This is just a bit closer up. There's one feature on the landscape here that tends to pop up in a lot of my pictures. There's these things called decade volcanoes. There's 16 of them on the planet, and they're big volcanoes that have a history of when they go bang in a very big way. And you're familiar with some of them, Rainier, Mauna Loa. Well, there's Uluwatu over here. This is Uluwatu. And this is our study village, the study place we were working. That thing's 100 metres taller than the highest peak in Australia, which is not necessarily saying much to people who have been to the Rockies, but still, for me, going along in a boat, and you just see this mountain going all the way up from the coast to the summit with a smoke trickling out the top, and you know that the entire continent of Australia would fit underneath that. It's kind of cool. So the village here, this image is already a couple of years old, but you're starting to see some of the scars of logging pushing through. The plantations have reached to here, but I don't think it'll be long before they jump across. But the village itself, until the latest logging tracks a couple of years ago, was completely isolated. No roads, there's no running water, no power. There is cell service. If you paddle your canoe off the reef and out a little bit, you can get cell service. And we had to do that one night. Well, I didn't do it, but there's one TV in the village at the school, and on State of Origin, which is not quite Super Bowl, but kind of, we couldn't get the TV to work. And so someone had to go and ring the teacher who was on holidays to find out how to get the TV to work so he could watch the game. The whole village on the grass in front of the school. But when we stay there, this is this lodge that was built at the village for guests to come and stay in. So it's remote, it's basic, but we actually get treated quite luxuriously. Flush toilets, hot water if you remember to turn the switch on. 3 nice meals a day cooked and put in front of you. It's not tough work by any means. The village itself, as I've mentioned before, is very much reliant on local natural resources. Not entirely. There's, I think, maybe for a village of 200 people, I think there might be 3 or 4 operating boats, long boats, outboards. And they'll use these to take the kids up and down hours boat ride down the coast to high school, for instance. But the lodge itself creates direct employment in the village. So all the guides, all the people running the lodge, the staff, everyone are from the village. And any visitors that come, part of their fee goes straight into a community account. For instance, they used it to put a tin roof on the church, put gutters on it, and some rain tanks to, instead of having to paddle your canoe across the bay every day to refill from the spring. And yeah, it's a pretty special place. There's some crocodile story here, but I might tell you that one later. Yeah. And the people there, as well as running the lodge, got very involved with the research as well. So Freddie here, very keen. He would, whenever occasionally one of these fish might get retained by sport fishers, he would collect the samples we wanted out of it. They would go out and collect other data we were interested in as well. So the fishery, what brings people there? There's that mountain again. Well, being a tropical island, this is about 250 miles south of the equator, right up there in the tropics, coral reefs right to the coast, and then dropping very steeply into very deep cobalt blue water. So naturally enough, there's a lot of fishing species, a lot of options there. You notice in this, all these people are in the little river boats. These are just the boats you putt around between the lodge and the river, and you can catch these things in between the lodge and the river. But as a lot of people would know, most of these species are very widespread. So they're a very attractive target, but that's not what brings people there. It's the rivers and the fish in these rivers that are the real draw card. So people will be fishing out of a boat like this, casting gear, throwing lures, targeting fallen trees, these big rainforests. Some of these trees are over 50 metres long. And these fish are lurking in the shadows under these sort of spots. And the key targets are 2 snapper species, Lajanus goldii, the New Guinea black bass, and Lajanus fusescens, the spot-tailed bass. So these are, same genus as red snapper and mangrove snapper here in the Gulf. And while those ones are the typical, these things get big. They get very big. The current world record for the black bass is 50 pounds. And this is a 50-pound snapper caught on casting gear from under a giant fallen tree in a flowing jungle river. They're pretty impressive. They hit a lot of surface lures too. You can get them up on the top. And they get very big. And so both species, you've got to always take it with a grain of salt when your most reliable source of information, fishermen. But I'm assured that they do get a lot bigger than what people manage to catch, because naturally, even small ones, a lot of the time, you've got very little chance of catching them, because where you hook them, they just bust you off into the logs straight away. But stories of village people carrying, one of these things that's 4 feet long and probably weighs 70 pounds on a log, on a branch, carrying it back to camp. So yeah, they get big, and I think for anyone who has any interest in fishing, the attraction's fairly obvious. And it's primarily a catch and release sport fishery. So virtually every one of these things that gets caught gets released again. They're snappers. They taste very good. The guides are quite happy if a group of anglers would like to taste one. They've got no problem keeping one or two smaller fish so that people can try it. But as you saw from the other pictures, there's no shortage of seafood to be had when you're staying in this sort of place. So most of them are released. And this is where we came in. They said, well, this is happening. We want to know what's already known about these species and what do we need to understand to try and manage this industry as it grows so that we can actually get maximum benefit for those people. So at the start of the project, we did fairly comprehensive searches of the typical databases, and using Latjanus goldii and pretty much every variation of how that could be misspelled, we came up with a total of 11 records. That's it, 11. And most of those were simply a species list. And some of those were Latjanus goldii, but it was probably misidentified, is what they've said. So in terms of actual ecological knowledge in any sort of literature, it's nothing there. So it's a pretty exciting project to get to work on. We know from the global distribution, when you go on FishBase, it says that the black bass occurs between Port Moresby and the mouth of the Fly River in the Gulf of Papua. So we did a bit of digging. I mean, you might already guess that we were catching them up here. We did a bit of digging and found that there's reliable reports, published papers, grey literature, other things that show that they extend at least around the island of New Guinea and these surrounding outer islands. We also found a report from a dive journal saying that they'd seen first record of one up here. And in our paper in 2016, we said, well, that's 3,000 kilometres from the nearest record. We think it's a mis-ID. I mean, these coral reefs, this is the coral triangle here. They're very diverse. We've got over 20 species of Latjanus in our study site alone. So we dismissed it. Since then, we've found fishing websites showing they look like black bass. Until someone does genetic testing, we can't be sure, but in the Philippines and out here in Kalimantan. So I'd apologise to those authors in that it looks much more feasible that they may have actually seen a black bass on the reef. That's in Iriomote Island, southern Japan. But in a global sense, it's still a relatively restricted distribution. The spot tail is a bit more widely distributed from mainland China right down through and extending out to the Solomons. However, when you think about the population density, human population density in this area, it's not surprising that we don't really find any reports of large fish of these sorts throughout most of that range. There's probably plenty in this part of the world, West Papua, but that is not a place you can really go visit very easily. Papua New Guinea is really the place where you can get these two species, healthy populations and big fish in an area where you can jump on a plane and go visit. So what we were interested in, as I said, this is an interdisciplinary project. I'm not going to talk to you about social science. I'll leave that to the experts. But we had social scientists, economists, but also a team of people, including myself, looking at the biology and ecology of the target species. So we were interested in where do these things move through their lives, particularly in a region where there is such large-scale impacts happening on catchments. We figure it's pretty important to know where each life stage goes and what resources they use, because you might manage the fishery well, but if you destroy the nursery habitat, everything's going to fall over. And we did some basic biology, age growth, and some diet study as well. Some of this has just been published in terms of the life cycle movements, and I'll just go through some of that now. There is a little bit of data in this presentation. It's not all just photos. So we used otolith chemistry, otolith microchemistry, to ask questions about the broad scale movement. So the general principle, if anyone's not familiar, is that as the otolith, the bone inside the skull of the fish, is laid down through its life, it incorporates some of the chemicals from the water into that bone. And so the chemical composition of the otolith itself tells you something about the chemical composition of the water that the fish was living in at that time. And so you get a record throughout the life of the fish. We're particularly interested in using strontium and barium. This is very widely used, pretty clearly established to show the greatest variation across gradients of salinity and turbidity. In other words, just perfect for if your question is, how much do these fish move between marine, brackish and fresh waters during different times in their life? As people in this room can tell you, the best approach, an approach that reviewers usually request, is to go out and collect water samples of your site so that you can relate the water chemistry directly to your otoliths. There's some various reasons I've put forward for why we didn't do that. I'd have to say probably one of them is that you can't collect a water sample while you're holding a fishing rod in your hand. And so instead, we went and collected a whole bunch of reef-resident lachanids from the coastal reefs and some freshwater fish. Well, in New Britain, freshwater fish, most of these are diadromous. It's a fairly young island. It's still rapidly changing. The elevation of mountains changes in some weeks. Elevation of reefs change. There's reefs there that you can identify all the species on that coral reef, but it's now 6 feet above the ocean because it just got shoved up during a minor earthquake. But anyway, we collected some freshwater fish, or fish living in a place where we're confident they would have been resident for long enough that their outer otolith chemistry would reflect what a freshwater fish should look like. And so we used these as our references. And essentially we're asking with the chemistry, during the lives, do these things, occasionally look like they're marine fish, occasionally freshwater, or brackish in between? And when we measure strontium and calcium, the message from this whole mess is that all of our reef fish had elevated strontium. Variable, but elevated. And all of our freshwater fish had very low strontium. And this is exactly what would be predicted from the literature, but you need to test to make sure that your local environment actually works. And these values are actually quite well separated. So we were then able to say if we were able to classify our fish based on how they related to these sort of boundaries of habitat. We included a couple other species, the spot-tailed, the black bass, but there's two other snappers. There's a lot of other snappers, but two that are also quite important in the fishery there. The mangrove jack and the fingermark. These are much more widespread, though, throughout the Indo-Pacific. So while they're highly prized, they're not unique to that area. But we looked at those because something more is known about the ecology of these species. And we classified each individual based on its profile. So if their profile looked like this down in the freshwater, we said that was a freshwater resident. If they showed some indication of low salinity, we called them brackish transients. Probably the interesting one is what we termed seascape migrants. These are fish that during their lives made these stepped movements through the seascape. So this would be, fairly significant ontogenetic habitat shifts between salty parts of estuaries, brackish and freshwater. Transients and things that had constantly high we classify as marine resident or saline resident. So these are the proportions. The main thing to get from this is that you see there's a gradient. So the spot-tailed bass is really very freshwater fish. It will move into brackish and move across the seascape, especially as they get larger, but the bulk of them are found in freshwater. The black bass is a bit more centred on the estuary, but it will move widely up and down between the ocean and the fresh. And the mangrove jack, much more in the marine part of those estuaries. And the fingermark, we didn't get many of them, but they're very much a coastal or river mouth fish. The other thing is these migrants. when you look at where they spent most of those time, most of that time in their lives, that also matches this general gradient. So there might be, people like to talk about niche petitioning and this sort of thing, but there's a gradient in dominance of where these things are. You can find all of them sitting on the one fallen tree in the river, but overall this is what the chemistry suggests they do. The other thing we got from the chemistry, doing transects from edge to edge across the otolith through the core, where we hit the core nicely, is pretty much clarifies these have marine larvae. So on one hand, I go, yeah, that's great. We found something brand new that Latjanus goldii and Latjanus fusescens have marine larvae, which is kind of cool, except every other Latjanus on the planet has marine larvae. So, I won't pat myself on the back too hard for that one. But we weren't sure. So I'll go through the acoustic tracking and the video together because their results kind of complement the chemistry. The acoustic tracking, I guess this is really just a brag. This is the biggest fish that we put a tag in. I caught it. So that's about 2 inches shy of 3 feet long and about 30 pounds. We didn't weigh it because we're trying to get an acoustic tag in it and get it back in the water nice and happy and healthy. But you catch the fish, that's the tough part of the job. Knock the fish out according to your IACUC protocols. You operate and implant the acoustic transmitter, a tag that pings out a unique number, and when it's within range of the listening stations that we deploy, the receiver records that number, and we can then use those to track where fish have moved. Once the surgery's over, we transfer it immediately into a recovery cage on the side of the boat, let them regain equilibrium, wait till they're swimming strongly, and then just drop the gate open and they swim out. And for various logistical reasons, our original plan was just buy a few receivers, a few tags, and go and check it, because the few conventional tagging studies of these fish had never turned up any recaptures. And the concern is these things die if you handle them like that. So our original plan, acoustics gear, people here know, it's expensive. The plan was do that, but for various reasons, especially working in very remote places, some of our trips got delayed. Well, anyway, it was sort of on my head that we ended up buying the full $100,000 of acoustics and going and deploying it in one hit over a month. And I spent quite a few nights that month sitting there going, I wonder how I word it if I have to tell the fisheries that for $100,000 they've learnt that you can't tag black bass. But they survived very well. We had excellent survival and long-term tracking. The deploying the receivers and maintaining the receiver array has been the biggest challenge of this entire project. All the other parts of the work, you have your challenges, but this is the big one. This is the very, very, very first prototype. We modified it a little bit. But essentially, equipment security is a problem anywhere. If you go and stick a big flag over the top of your expensive field gear, it only takes one person to decide that they want to see what's inside using a brick, and you've lost it. But we came up with this idea so that we could submerge them. And so we needed actually quite deep water for our array because we relied on these staying far enough below the surface that you wouldn't see them. And look, I got more sympathy. We lost quite a few receivers for various reasons. And I have quite a bit more sympathy up here when you think that a big chunk of steel and probably more importantly 20 feet of rope and some chain is a pretty rare and precious resource. So on those days, we would submerge them and they'd be well below anything we could see, but occasionally these rivers go crystal clear and you can see a leaf in 20 feet of water on the bottom. And someone paddling along. Oops. Is that still on? Yep. So anyway, we lost a few. The other one, we got a few back, but they were a long way from where we put them. And we find them, just you're sort of almost given up and you're throwing the grappling hook out and just driving in the boat, and then suddenly you jag the thing and pull it up 60 yards from where you left it. And we were getting them back and the floats had all these funny big holes in them, and we just like big puncture holes right through them. And so It turns out that having a float that's sort of sitting there wafting in the water like this doesn't work. So our second version, we removed the floats, removed the croc attractors, and just submerged them. But then they started burying very quickly. And it was okay if we got back every 3 or 4 months and pulled them up and put them back down again. But, you know, one of our trips, we got delayed 6 months because the police were shooting students that were protesting in Port Moresby, and they wouldn't let us go back up. So, yeah, maintaining that array was the biggest challenge. I did say there would be some croc story in here. And the approach you take, you put those receivers, spread them along the rivers of interest, limited in our case by places where they're not going to just get barreled out of the road as soon as a bit of a flow comes through and sends another 200-foot log down the river. But we spread them between offshore areas, like here there's coral reefs right in front of this river, through the estuaries and all the way up into freshwater, and across this sort of interconnected mangrove swamp and big river here. I'll go through the camera stuff before I sort of show you the integrated results from those two. So the underwater video work, the good bit about that is compared to a lot of other techniques, you actually see what's where and exactly what it is they're up to. And we're also able to identify the nursery habitats or the location of these little guys that you don't catch or you don't find using other means. So using the three approaches together has some benefits, especially because each one on its own doesn't really give you a completely conclusive answer. So the good thing about otolith chemistry is you get a lifelong record of the sort of water chemistry that fish has been in from when it was hatched right through until when you caught it. But using that alone, it can be difficult to distinguish a fish that's moving through different water chemistries from a stationary fish experiencing variable environment around it, which is just what you might expect of a fish living in a dynamic estuarine area that can be freshwater one day and saltwater the next. The movement patterns you get from acoustics are very detailed. They're very specific. You have an ID for an individual fish and you know when it's within range of one of your particular receivers, and you can map its movement up and down these rivers. But that's limited to the number of fish you can tag, how well you can keep your array in the water and the spatial extent of it, and the battery life of the tags, which is the ones we're using are about 3 years, which is pretty good, but still only a small part of the life cycle. And the video gives you information that, at least at the scale we applied these other tools, you just can't get from those. So specific habitat information, exactly what type of habitats within those areas are these fish using. But on its own, spatial patterns of distribution don't necessarily infer movement. You can get differential mortality and other things like that. But by combining these, we were able to overcome this. So for instance, the movements that we inferred from the chemistry were made by individual tagged fish. So the same scale. We look at the chemistry and said, it looks like these black bass live in the estuary, but will go out to sea and will go well into fresh water. And the tagged fish did just that. And then the distribution of them matched as well, the different sizes or size classes. And then the lifelong record that you get from the otolith chemistry meshes quite nicely to a very different scale, but to the shorter term record you get from the acoustics. So you can put the two together to kind of fill in the gaps in the other technique. And similarly, both of these approaches can help confirm that what you're assuming is distribution patterns and movement, what you're assuming is movement of different life stages really is that, because it's what you're seeing in the other two techniques. So what did we learn from all of that? Well, the spot tail has marine larvae. Pat myself on the back again for that one. They recruit pretty much straight up into fresh water. And in the freshwater area, the video surveys showed that they are very widespread. You pretty much go, if they're in that river, you go to any branch, log, anything from a small branch up to a giant tree, and they're there. And they're on the same habitats as the really big adults, maybe using them a little bit differently, but They're widespread, so they don't really seem to have a very specialised nursery habitat other than structure, which in rainforest rivers there's plenty of. They grow and spend quite a bit of time up there, and at some time around the age of sexual maturity, they tend to show a bit of a downstream shift. So those seascape migrants, the bigger ones, typically move from fresh to more salty areas. And the acoustic tracking showed that they will make movements into the mouth of these estuaries and even offshore. We detected them on the reef outside the mouth, and there's no way that those transmitters can ping through the river mouth to get out there. So one interesting thing, this is really, as far as I can gather, the extreme of freshwater occupation amongst the whole family. We think of tropical snappers, they're coral reef fish. We know that a lot of species will spend parts of their lives up in freshwater, like mangrove snapper here, but these things, 50-pound adults living under logs in flowing rainforest streams, kilometres up from the ocean. One thing we don't know is the spawning site. All of these movements give hints about, maybe that movement is going out there to spawn, or maybe this, but we haven't been able to pin that down. One thing is we can't work there during the wet season. The rivers, those big trees, each year you go back, they're different trees. These rivers, when they flood, they really flood. The mountains up here might get 10, 12 metres of rain a year. It's huge rainfall. And it seems possible, the bulk of the spawning, based on their gonad condition through the year when we are there, that they're probably spawning during the summer mostly. They are active at other times. And we have found large aggregations on trees, on snags, fallen trees, on the salt wedge at the top of the estuary. And they showed no interest in feeding. So when there's a whole bunch of mature-sized fish sort of milling around, no interest. Well, I couldn't catch them, so surely they weren't feeding. We didn't have any dynamite, so we couldn't figure out what they were doing. We suspect those might be aggregations, but whether they spawn there or whether they shoot out the mouth of the river and spawn at night and come back, we don't know. There's interest in that because especially, as logging roads push in onto the beaches and people can suddenly bring trucks, people who don't live there can bring trucks in and maybe stick gill nets across river mouths and things like that, or use poison rope, which is a vine that grows there that'll asphyxiate fish when you dump it in the water. So we want to know more about when and where these things actually spawn. The black bass, marine larvae, they migrate. They have a much more limited, at least what we've observed so far. We're only looking at a limited part of the range and the seascape where we could actually get to. But the only place we find the little guys is in about a metre of water along grassy riverbanks, right at the upper tidal limits. It's fresh water mostly, and especially where you have a little bit of extra structure like a branch in amongst the grass. So quite limited. Between this big and the sort of when they start getting captured in the fishery or being abundant, which is about 18 inches, we don't know where they are. And using the angling approaches, you catch plenty of the other snapper species that are this big, small ones, but we don't catch these. So maybe they're going further upstream. The chemistry suggests this is maybe what they're doing, but it's a little ambiguous. But from the time they reach the fishery, they're back down in the estuarine areas or the lower freshwater. And like the spot tail, as they get bigger, they make these downstream movements into the more salty areas. But these tend to end up in the lower part of the estuary, at least in this region. And with the acoustics, it also shows they make very regular wide-ranging movements. We had half our black bass, making regular movements outside the river mouth to coastal waters and upstream. And, individuals, that big one, swimming from the estuary at least 10 kilometres upstream, and these aren't big rivers by any means, 10 k's upstream across shallow sandbanks in a day or two, spending a few days up there doing who knows what, and then coming back again. We don't know about spawning site for these ones either. It's still unclear, and again, their gonad condition really suggests that the peak spawning activity is going to be during the wet season when we can't get there. Another issue for these we haven't got to yet is among river movements. And this is important in an area where typically a community owns the river, or sometimes in some rivers more than one village. But if the populations in each river are pretty much staying put from when they recruit, then an individual community can decide how to manage their river. If that population's moving and mixing amongst multiple rivers, sort of effectively multiple jurisdictions, then you've got to get management that cooperates at a broader scale. How are we going on time? Anyway. Okay. A little bit on age and growth. One of the things, this just published last year, like a lot of snapper species, the size, each individual seems to have quite different growth rates or growth trajectories through life. So small individuals fixed size individual can be a very wide range of ages. So if you see the top one here, the sort of 15 to 18 inch black bass that people start catching were anywhere between 3 and 12 from what we could gather. And the spot tail is very similar. Now, this is quite common for a bunch of Lutjanids, especially as they get older. You can get really, two red snapper this big could be 20 years difference in age. And so what this means from a management point of view is that small fish could be very old fish. Now, a little fish could be either a new recruit to the fishery or it could be 10 or 12, 15 years old. Now, in an area where we don't know where they are, those younger ones, possibly upstream in areas that are getting mined and logged and plantation agriculture, you could have complete recruitment failure to that fishery for 10 years before you'd even notice that you're no longer catching little fish. And by that time, it's probably too late. So we wanted to know, can we age these things without killing them? Now, this is nothing new. I mean, the first aging of fish was done hundreds of years ago using fish scales. We looked at dorsal spines, and again, this is nothing new. Other people have done this, but you need to test if it's actually going to work with your species. So basically, the otolith here, showing the growth rings. See, you can see some of them sometimes. And when we section a dorsal spine of the same fish, we get the same age estimate. And when you plot this up, basically we've got the age count from the otolith and the age count from the spine. So any dot that falls on the line is a perfect agreement, and the dotted lines are 95% confidence of the otolith age. So the key message there is the dorsal spine is going to give you pretty much the same age estimate as the otolith. And instead of killing the fish, cracking its skull open and pulling the otoliths out, you take a pair of wire cutters and clip the spine off and let it go again. It doesn't require any specialised training or equipment. The guides can do it, community can do it. Easy to store them, you just stick them on the shelf until you're ready to send them off to the fisheries and get them processed. The food web stuff is in prep. We did diet studies on these things. They're snappers. They eat lots of crabs and crustaceans and fish. And the isotope work is being analysed now. So the other thing I wanted to talk about is the food webs we found up there. So one interesting thing in these rivers, the diversity is a bit lower than what I'm used to in nearby northeastern Australia. And I think these estuaries, they're quite young geologically, and they just don't have such a diverse fauna. But the interesting thing in those places is most of what you catch are predatory, whether they're this big or this big. The bulk of the species, you get some mullet, but you don't get a lot of detritivores. You get a few of these benthic invertebrate feeders, but not many. There's not many planktivores. You don't really get many planktivores when you've got a stream blasting out of the mountains. Most of the things are predatory, and yet so you're wondering, what are they all eating? Well, the locals would tell us about whitebait. The whitebait runs come in on the new moon. And we wanted to know what these were, and we were present for one of them. So this is just off the side of a boat up one of the rivers. I'll just pause it for a second. What you're looking at, all of this is fish. These are inch-long fish. This is a foamy, slimy scum of fish slime. There's that many of them there. And in fact, the slick that you see stretching off in the distance and all the way up the river, and you can't see it, but lining the entire other bank of the river is just like this. This river is full of these things. Coming into the river on a day like this, from a mile out to sea, you can smell this like rotten fish smell, and it is incredible. These are just these little inch-long fish that come flooding in their billions. Some of these events, I don't think billions is an exaggeration. It's an absolutely phenomenal biomass of things that just comes pouring in. And they don't come in every new moon. Sometimes they're there, sometimes you see a few, sometimes it's like this. And when they arrive, everything goes nuts on them. The fish, these bass, I mean, they're pretty aggressive fish, but they're not tuna. They think they are for a few days. I mean, the birds get into it, and the people from the village, they'll know the whitebait runs on. You go camp at the river mouth, brave all the midges that smash you the whole time, and just you see kids with a basket just scoop, and they've got pounds and pounds of these fish. I ate the fritters once that they made, the slightly undercooked fritters that were made with these fish that had been lying in the bottom of the boat for a few hours in the tropical heat. They're okay. Nothing's hatched yet anyway. But these are incredible, and it's pulses of prey like this coming in. So we wanted to know what they were. We collected some. These are post-larval cyclodyne gobies, cling gobies. So the adults are some pretty spectacular fish. There's a whole bunch of species through the Indo-Pacific. Some of these things, they're all all diadromous. The adults live up in the fresh water. Some of them live at altitude. They can climb over waterfalls to get up into the streams. The eggs hatch, they spawn, they put their eggs on these sort of flowing clear gravel beds, and they find rocks and spaces. There's fish around here that do that somewhere. Put eggs, in under the rocks. They hatch, they get swept out to sea for a few months, and then these post-larvae come flooding back in. And that pulse of energy, we suspect, might have a huge amount to do with the carrying capacity and the food webs in these rivers. But people here, there's fish here that do something like that, and what happens if you go and log that whole catchment and dump a whole bunch of mud into those gravel beds? So we've got some serious concerns about what this might mean for the ecology of these rivers, the carrying capacity and the fishery. And we only have a bit of anecdotal evidence from people further down the coast where the agriculture is, pretty much all-encompassing, that say, yeah, we don't get whitebait here anymore, but Grandad said we used to. That's about the extent of it at this point. But so, stepping back a bit for the bigger picture, it's a nice place, it's exotic. Can we learn anything for here? Or is there anything relevance to here? Well, I think there is. You can pretty much substitute any fishery resource. I think the Gulf Coast is a great example of places where people really do care about their fisheries resources. And to me, again, this sort of really shows how the sort of work I do links in with things that matter to people. And also, as fisheries management has moved from a focus on single species to try to encompass more ecosystem-based management and tie in some of these other fields as well, what this brought into sharp contrast for me is that a lot of the things really impacting these fisheries are external to what fisheries people have jurisdiction over. And so that's the direction we're moving, but it's a big task to try and tie these in. And again, while this was the PNG context of mining, logging and such, this applies equally, anywhere here along the Gulf Coast, that there's a lot of pressures on these systems that are from well outside a fisheries manager's jurisdiction. Maybe on a more positive, sciencey note, the other thing I'm pretty interested in is how does the function vary geographically? How do these life cycle movements vary geographically? We studied in one area of the range of these fish, and the seascape there is quite different even to, say, other parts of New Guinea with the black bass. So if we look at this, we were in pretty small coastal rivers compared to, say, the Fly River. That mine I showed you earlier is right there on the top of it. This is the biggest river in the country, and black bass have been found 1,000 kilometres upstream at the base of the Central Mountains. They've also been found, again, at least we've been shown photographs, pretty sure that's what they are, out on oil infrastructure in the Gulf of Papua. So the sort of questions I'm interested in there is, do the life cycle patterns we saw at this kind of scale, where you can swim from fresh water to a coral reef in about 20 minutes, do they match with the same sort of patterns here, or do these fish over here do something different? The mangrove jack we looked at this site really do, even though that seascape is compressed into a very small space, they showed the exact same sort of ontogenetic habitat movements that mangrove jack on the east coast of Australia do, even though in Australia those reefs are tens to 100 kilometres away from the estuary. And I think I'll wrap it up there. Thank you. Yeah.
How effective are your tags at penetrating thick tissue? Could your results be skewed by crocodile predation?
Right, so could this tag keep transmitting after a crocodile has swallowed the bass? It's possible, I think. However, when you look at the general patterns of the way the animal's moving, you know, if the animal sort of hung out on the bank next to that receiver for a long time. Although people have tagged crocodiles and show that crocodiles move around an awful lot more than we thought they do. It's possible, but no, I think most of what we're looking at is actually the fish. Also, you know, sharks. There's a lot of sharks in these rivers, bull sharks and such. And so certainly they would, you know, a fish recovering from surgery could be quite vulnerable to a shark. But we're pretty convinced, based on the movement patterns that we were seeing, that these are mostly we're not looking at predation. And there's in fact some new tags, I believe, that they're making that especially change their transmission when stomach acid dissolves the outer casing of it. They're probably a lot more expensive again. Yeah. So the first part of the question, estimating abundance, the video surveys were pretty comprehensive. Michael went and put out 1,000 video drops. And very quickly, we just record presence. But what you quickly build up is, what's the probability of finding a black bass in habitat X? And they're really common. The fisheries dependent, being angling, it won't work. One of the tagging trips, it's crazy, the guy that does the acoustic tagging doesn't like fishing. I mean, our job is go to New Guinea, stay in a lodge, and go catch these things that people pay $1,000 a day. And he doesn't like fishing. He's a shark person and a scuba diver. But anyway, the angling, we spent so much time at the rock bar at the top of this river. There's fish all over it. And he's sitting there going, that's not true, obviously. There's no fish in here. And then we got there one day and the water cleared up more than we'd ever seen it. And we're sitting in the boat, and me to these chairs away from us was more fish than we needed to tag in the entire study, and they're just looking at us. And we drifted everything, we tried everything. And occasionally you'll go there and you'll catch 50 fish. But mostly, you know, a lot of the times they're not interested. And so I think the angling catch rate as an indication of abundance is probably not so good. But certainly I think there's some opportunities. The other one would be something like pit tags or the coded wire tags. We were reluctant to go externally tagging fish because people are paying a lot of money for an experience. They're going to a remote wilderness location, and if every third fish they catch has got a bit of plastic hanging out of it, that could impact their enjoyment. So, but if in future I would have done some pit tagging. We did get some recaptures of our acoustically tagged fish, and I think we could have nailed down a few things like verifying the ages and such using pit tags. Yeah.
So we have a pretty popular root genus that we study here, and
I think I've heard of it. Yeah.
And we know now that they get quite old, upper 50 years. Do you have an idea of how old a root genus is? And also with your non-lethal aging, do you have issues aging some of the older ones that go into 15 years old?
Right, we didn't, but so the oldest fish we got was 18. They're not, by no means the biggest, and I would suspect these things are probably getting up around, well above 30 or more. The spines won't work for those bigger fish because of occlusion. So unlike an otolith, which never gets modified after it's laid down, the spine blood vessels start to grow up the middle of it, and it gradually dissolves away the inner growth rings. And in that picture I showed, you could see the first growth ring was almost obliterated. Our interest with that was really about the young fish, how old, or the small fish. Certainly, I don't think spines will work for the really big ones. And not many people keep the big ones, but we did put out a call through the person that runs the operation saying if you ever see someone with a big fish, here's how you get the otoliths, here's the pliers, make sure you get them. But so far, thankfully, most people don't kill the big ones at all, or not intentionally. Yeah.
With the freshwater fish, you use the profile otolith signature. Do you have any large freshwater adult fish you used for that, or was it all
No, they were. So the Coolia, it's a family called Coolidae. They're diadromous fish that spawn. They basically live in freshwater the whole time. They migrate down to the coastal water, spawn, and then go straight back. So we had, they're only this big, but they're adults. And those and then some small spot tails that we got a long way upstream and some other things, they all show the exact profile you'd expect, which is a very clear marine core going through a brackish estuary and then flattening straight off into fresh water. So they were pretty convincing, but that's a different family entirely, that freshwater one. So, yeah. On the other crocodile story, Gabby, one of our guides, a young guy in the village, it's not a good story. I feel like I need to get back to that picture though. Oh, come on. Where are they? Here he is. What? Excuse me. I do owe it to Gabby to bring his picture up. Go away. Go away. Come on, this is not a lecture. Yeah, Gabby here. This is son Martin. So I was sort of saying it's a pretty special life these guys have got there, but it's also tough. He was out with his wife collecting mud mussels, clams out of the mud in the mangroves, in the water, feeling around with your feet, and you find one, you dive down. He dove down, big swirl, he's gone. They never found him. Yeah, that's his little boy. He got eaten, and it happens. There's a lot of malaria in the village. I managed to somehow not get it despite spending a few months there. So there's a lot of benefits of the lifestyle those guys have got, but they are pretty isolated and they do have to take a lot of risks to put food on the table sometimes. Yeah. So I should have saved the happy crocodile story, shouldn't I? Anyway. All right, thank you.